Good morning, dear students. Today I'm going to continue the rest of the lesson, and we are into another major topic called post-fertilization. Okay, let's see the previous class. I discussed about the fertilization in previous session. You can see there are few things I would really like to revise, like double fertilization, the most important one that we discussed yesterday. Double fertilization is the fusion of, like, there are two fertilizations that happen in plant body. One is syngamy, other one is called triple fusion. I even said syngamy. So syngamy is fusion of one male gamete with egg cell, which is N condition, and N condition will lead to zygote. Same way triple fusion of one male gamete with a polar nuclei lead to 3N condition, triploid cell called peck, and nucleus is called pen. So that's why it is called double fertilization. So up until this, we completed our two major steps. Those are pre-fertilization, gametogenesis, fertilization, and then comes the post-fertilization. Let's see what happens exactly soon after the fertilization. Thus, the events that happen soon after fertilization. See, we have an embryo sac which has undergone fertilization. Remember, the things that are present apart from these egg and central cell, like antipodals. These antipodals and the synergids start degenerating, and then nobody knows what exactly antipodals meant for, but they are present over there in the embryo sac. If you see the embryo sac, you can see the rest of the parts, like antipodals and synergids, will get degenerated, but the only cells will remain are two cells. Those are they are called zygote. And zygote, apart from zygote, there is another cell called central or secondary nucleus, which is in 3N condition. This we already discussed in our previous class. So 2N and 3N condition cells which remain, and the rest of the cells like synergids and antipodals start degenerating. So let's see what happens exactly soon after the fertilization. Let's see the fate of the central cell. I'll take the same page right over here. I'll start with the what is the purpose of double fertilization. In that, one of the cell called central cell or central nucleus need to be what is the purpose being present right in the embryo sac. Remember, these 3N condition, whether it is a 3N condition, central cell or a zygote, obviously they will undergo one specific cell division called mitosis. So no more meiosis. That means meiosis is only confined for production of gametes only. That we already discussed in the gametogenesis. But soon after the production of zygote and PN or primary endosperm cell, they'll start undergoing mitosis. This we discussed in our class 11 standard also. If you look at this picture right over here, this is a chapter called plant growth and development. In that we already discussed that soon after zygote formation, zygote will have to be undergoing two major mitotic divisions. One is called geometric, if you remember, geometric, and as well as arithmetic division. Zygote, primarily, it starts dividing by geometric phase of its cell division, where each and every cell will undergo or participate in cell division, resulting in many number of cells, daughter cells we can say. Soon after that, geometric cell division, then the cells start undergoing differentiation, where cells stop dividing, some cells stop dividing, and the cells will become mature, and the cell division that will take place is arithmetic rate of growth rate, you can say. So the same thing happened for both for the PEC, PEC cell, primary endosperm cell, and the zygote also. First we'll discuss about PEC. What is the future or the fate of the PEC? This, I'll write here, the main purpose of it is to do one major thing. The PEC, the purpose of having the triple fusion in the plant body is to do one major thing, is to provide nourishment. Nourishment for what? For whom? Provide nourishment for the development of zygote. You know that zygote is the actual cell that will develop into an embryo, and embryo develop into seed. So for the whole embryogenesis process, from single zygote to become seed, there must be some tissue providing nourishment, and that cell or that tissue will be the endosperm. The endosperm is the tissue that will provide nourishment to the complete embryogenesis process. Unlike human beings, human beings have only one zygote. We have something, other developmental stages which will be guided or nourished by some tissue called placenta. So this, in humans or any other animals, only one cell division, fertilization takes place, that is syngamy. So whereas since the nourishment tissue in the humans is provided by placenta, but there must be some tissue providing the nourishment for the whole embryogenesis process. So here comes the hero. Who is that hero? Endosperm. If you look back, even if you look back from the beginning of our plant reproduction studies, each and every stage have their own a nourishment tissue being present. If you take megaspore mother cell, MMC, or microspore mother cell, everything is been completely nourished. Suppose if you take megaspore mother cell is nourished by tapetum, and microspore mother cell is part of the microspore mother cell is nourished by nucellus. If you go soon after that, even after fertilization, there must be some tissue which must be providing nourishment for both the zygote and endosperm. Okay. Now, if you take the classical example that is given in our textbook, look at this thing, whole thing. I'll tell you clear way what exactly is this. This endosperm is like a tissue that guides the zygote complete embryogenesis process. In our textbook, they given a perfect example. This example is the coconut, the huge coconut that we see. I draw a rough picture of coconut. This is the coconut, green one. If you cut open, you can see, we already discussed in our 11th standard also. You can see there is a shell, the hard structure, which consisting of white tissue, which is commonly called kernel. So kernel is the white tissue, and you can see the coconut milk or coconut water present. Actually, it is not actually the water part. If you take very tender coconut, tender means like freshly plucked, and compare it with the mature coconut, you can see there will be a lot of difference. If you observe the tender coconut, cut open and see that inside that there are plenty of water will be present. But if you see the mature one, you can see there will be no water, but it is completely filled with the kernel, white tissue, and you don't find water in it. What exactly is that? Why the difference between tender coconut with the mature coconut? It's simple. The same thing happened right over here, the whole thing, whatever I discussed. Remember, the white kernel that you are observing is actually an endosperm. You may think that this white kernel is the actual embryo, but that is completely wrong. The white kernel that you are observing is not an embryo. It is just an endosperm. You may ask me, sir, then where exactly it will be present? It is present right exactly here as a green tissue, and this is embryo. Somewhere inside that tusk, there is a green embryo growing. This is actually the embryo surrounded by the endosperm. Now let's see the evolution of the tender to mature endosperm. Now if you take the same circle in a big picture like this circle, here the endosperm cell, the initial endosperm cell that we discussed right over here, PEC, soon after fertilization start dividing. That means imagine this whole thing as a cell. So suppose I'm drawing the same here. Suppose this is a big complete circle of the coconut. Now then remember, the PEC will start dividing. That means karyokinesis occur, and there will be production of many number of nuclei. I already said karyokinesis is the division of nucleus. Division of nucleus led to the formation of many nuclei that it will increase in the number of nuclei. So this nuclei that you are observing inside the coconut is the actual it is made up of number of nuclei, which is the one which you are drinking, that water. The water is the one which is made up of number of nuclei. Because you may ask me, sir, then what exactly could be the kernel? This liquid that you are observing is the nothing but free nuclear endosperm. Free nuclear endosperm is nothing but number of nuclei that is looking like a water without having any cell membrane around it. So this free nuclear endosperm without cell membrane, that means each and every nuclei appear like a water. That is the nucleus of the endosperm that we are actually drinking. And if you go to south, coconut milk or coconut water is like in high demand. Soon after the free nuclear endosperm, the nuclei is formed. The tender coconut water is high. But slowly the tender coconut start becoming mature. You can see there will be no water. What is the cause of it? The free nuclear endosperm, that means the nuclei start developing its own membrane around it. Slowly the nuclei become the solid part, that means the kernel, the white part. That kernel or that white part is nothing but the water, that which we call free nuclear nuclei, the nuclei being surrounded by cell membrane, giving them the solid nature, which we call cellular endosperm. Thus the cellular endosperm becomes solidified and slowly becoming as a white tissue later, which we eat, or we can use it for preparing some delicacies in kitchen. So that is what the difference between free nuclear endosperm and cellular endosperm. Water is nothing but free nuclear endosperm made up of many nuclei before the cell wall or cell membrane formation, and when the tender coconut matures, each and every free nuclei surrounded by cell membrane to form the kernel. Kernel is actually called as cellular endosperm. So there are two endosperms. So these two, whether it is a free nuclear endosperm or cellular endosperm, will provide nourishment to the growing embryo or the zygote to become as a seed. Okay, so this is about endosperm. There is one specific thing that has been given. You can see this is the free nuclear endosperm and the cellular endosperm. That is neatly explained in today's class. There are two types of endosperms are present. One, there are two types of seeds are there. One type of seed is called endospermic seed, and the other type of seeds are called non-endospermic seed. This we'll discuss even later also. And there are other names to it. Endospermic seeds are called albuminous seeds, and this is called exalbuminous seeds. The reason why is, what could be that? The name itself tells endospermic seed means the seeds which even after embryogenesis, like usually in order to understand this, I have to be telling this. Suppose a zygote developing into a seed called non-endospermic seed. So during the course of embryogenesis, the whole endosperm is utilized so that the zygote develop into seed, and the seed no longer consisting of any endosperm being present around it. Say the example of a pea, groundnut or beans. So in this type of seeds, endosperm is completely absent. That means this completely, during the process of embryogenesis, zygote developing into embryo, the whole embryogenesis will suck up or consume each and every endosperm to develop into a complete seed lacking the endosperm. Whereas if you see the endospermic seed, the zygote developing into a complete seed still holds some tissue of endosperm being present attached to the seed. So not only during the course of development of embryogenesis, even after this embryogenesis, the endosperm still remain. So here we can take an example of maize seed. We'll discuss that even after embryogenesis also. Maize and castor are the best example for endospermic seeds. Okay, we'll discuss this thing soon or later. Okay, so please make sure that you cover the rest of the topics also. Thank you and have a nice day.